The plague is caused by a bacteria called Yersinia pestis, and most of the time humans get it from a flea bite. Back when plague was rampant and there was no treatment for it, plague could get into people's blood and it could turn their limbs black.
Did you know the Black Death killed millions and changed history forever? For hundreds of years, people wondered where this deadly plague really started. Now, thanks to amazing new science, researchers have found the true origin by studying the DNA of people who lived through it. This discovery finally solves a big mystery and gives us a clearer picture of one of the darkest times in human history. In case it happens again, is humanity ready for another deadly pandemic? The Plague of the Black Death. In the mid-14th century, Europe was forever changed by a mysterious and deadly force. The Black Death swept across the continent with brutal speed,
And most enduringly, the Black Death, this mysterious plague,
leaving devastation in its wake. It was more than just a disease. It was a catastrophe that reshaped societies, economies, and even the course of history itself. Between 1347 and 1351, this plague wiped out an estimated 25 to 50 million people, roughly one-third of Europe's population at the time. The sheer scale of death sparked fear and confusion, with no clear understanding of how the disease spread or where it began. The symptoms were horrifying. Victims suffered from high fevers, chills, vomiting, and painful swellings known as buboes, large, pus-filled lumps in the lymph nodes, especially in the armpits and groin. These buboes gave the disease its alternate name, the bubonic plague. But not all cases looked the same. Some suffered from septicemic plague, which infected the blood directly, while others had pneumonic plague, attacking the lungs and making the disease transmissible through coughing. The rapid progression and high fatality rate terrified communities as people watched neighbors, friends, and family members fall ill and die within days. The plague didn't respect social class. Kings and peasants alike were victims. Cities like Florence, London, Paris, and Venice saw their populations decimated, with entire neighborhoods abandoned as people fled the disease or succumbed to it. The death toll overwhelmed burial grounds, and mass graves became common. Social order broke down in many places. Labor shortages drove wages up, changing the economic balance, and eventually contributing to the decline of feudalism. Meanwhile, religious institutions were both sought for comfort and blamed for the calamity, fueling persecution, particularly of minority groups like Jewish communities. For centuries, historians and scientists have debated where the Black Death actually began. Some early theories placed its origin in Europe itself, suggesting the disease emerged from local rodent populations. Others looked further east, to Central Asia or China, regions linked by expanding trade routes. The most widely accepted idea became the Silk Road theory, which proposed that the plague traveled from Asia into Europe along the bustling trade routes connecting east and west. Fleas infesting rats aboard merchant caravans and ships were thought to be the carriers, spreading the infection from one city to another. But these explanations, while plausible, were based on circumstantial evidence, records of outbreaks, trade patterns, and historical accounts, but lacked direct proof. The true origin remained shrouded in mystery, a puzzle missing its crucial piece. How could researchers definitively trace the plague's roots when the evidence was so old and scattered? Enter modern science with a revolutionary tool, ancient DNA analysis. By examining the genetic material preserved in the remains of plague victims, scientists could now read the biological history of the disease itself. This approach offered a way to move beyond speculation and find concrete answers buried beneath centuries of soil. What exactly is ancient DNA, and how do scientists use it to uncover secrets from the past? How did this technique finally shine a light on the real origin of the Black Death? Scientific methods for studying ancient diseases. How do scientists peer back hundreds, even thousands of years to unlock the secrets of diseases long gone? The key lies in the remarkable science of ancient DNA, or aDNA. This technique allows researchers to extract tiny fragments of genetic material from the remains of people, animals, or pathogens buried deep underground. It's a bit like finding a fragile, faded manuscript hidden in a dusty attic and carefully reading the words before they disappear completely. Ancient DNA research began gaining ground in the late 20th century, as improvements in DNA sequencing technology made it possible to decode genetic material from old bones and teeth. But studying aDNA is no simple task. DNA breaks down over time, affected by heat, moisture, and microbes. Often, what scientists find is fragmented and damaged. Contamination is a constant risk too, as modern DNA from researchers or the environment can mix with ancient samples, leading to false results. To overcome this, laboratories have developed strict protocols. Samples are handled in clean rooms, and special chemical treatments are used to isolate genuine ancient DNA. When it comes to studying the Black Death, scientists focus on extracting DNA not just from human remains, but from the plague bacterium itself, Yersinia pestis. This bacterium is the real culprit behind the Black Death and its deadly relatives. The trick is finding preserved traces of Yersinia pestis DNA inside the victim's bones or teeth, which can survive for centuries if conditions are right. Once the DNA is extracted, researchers use advanced sequencing machines to read the genetic code, millions of letters long, and then compare it to known sequences from modern strains of the bacterium. By analyzing differences and mutations, scientists build a genetic family tree that reveals how the pathogen evolved over time and where it may have originated. This approach is known as phylogenetics. This method provides more than just a snapshot. It allows scientists to track the plague's evolution, identify branches of the bacterium related to historic outbreaks, and estimate when key mutations occurred. The combination of archaeology, genetics, and bioinformatics has transformed the study of ancient diseases from guesswork into precise science. In recent years, multiple international teams have applied these techniques to plague victims from various burial sites, spanning Europe and Asia. The DNA extracted from these sites offered critical clues, linking the devastating 14th century outbreaks to earlier strains found in Central Asia. These findings have begun to piece together the plague's journey from its place of origin to the wider world. Discovery of the Black Death's origin through ancient DNA. For centuries, the origin of the Black Death remained one of history's most puzzling mysteries. Thanks to advances in ancient DNA research, scientists have now uncovered direct genetic evidence tracing the source of the pandemic to a very specific region and time. This discovery didn't happen overnight. It was the result of painstaking analysis, international collaboration, and the fusion of archaeology with cutting-edge genetics. The breakthrough began when researchers extracted DNA from the teeth and bones of plague victims buried in mass graves scattered across Europe and Asia. These remains, some dating back to the 14th century, held the key to unlocking the plague's past. By isolating fragments of Yersinia pestis DNA preserved within, scientists could compare them with modern and ancient strains to map the pathogen's evolutionary tree. One landmark study, published in recent years, analyzed DNA from remains found in a burial site near Lake Issyk-Kul in the Central Asian steppe region. This area, spanning modern-day Kyrgyzstan and Kazakhstan, had long been suspected as a possible origin due to its location along early trade routes and historical accounts of plague outbreaks. The ancient DNA confirmed these suspicions, showing that the strain of Yersinia pestis present there was the common ancestor of all strains responsible for the European Black Death. Genetic sequencing revealed that the bacterium had evolved in this region around the early 14th century, just decades before the first recorded outbreak in Europe. This timeline aligns perfectly with historical records of the plague spreading along the Silk Road, transmitted by fleas hitching rides on rodents traveling with merchants. The DNA evidence thus provided a direct biological link between Central Asia and the devastating pandemic that later engulfed Europe. Further studies reinforced these findings. DNA extracted from remains in other Central Asian sites showed related strains, painting a clear picture. The Black Death originated in the Central Asian steppe, where the bacterium had circulated in rodent populations for centuries. From there, it jumped to humans and began its deadly march westward. This discovery sheds new light on how the plague spread. The role of trade routes like the Silk Road was crucial, acting as the highways for rats and fleas to carry Yersinia pestis over vast distances. Human movement, from merchants to armies, facilitated the jump across continents. The DNA evidence transformed theories into facts, revealing a precise origin point supported by genetic data. Moreover, comparisons between ancient and modern plague strains showed how the bacterium evolved to become more or less virulent over time. The strain responsible for the Black Death was particularly deadly, with genetic markers linked to high transmission rates and fatality. Understanding these genetic factors helps explain why the pandemic was so catastrophic and why later outbreaks, such as the third pandemic in the 19th century, had different characteristics. Uncovering the real origin of the Black Death also helps historians and epidemiologists understand the interplay between environment, human activity, and disease evolution. The Central Asian steppe, with its diverse rodent populations and fluctuating climate, provided an ideal setting for Yersinia pestis to mutate and jump species. Human trade and migration then turned a regional disease into a global catastrophe. This new genetic evidence closes a long chapter of speculation, replacing it with a clear, factual narrative. It demonstrates how ancient DNA can reach back through the centuries to tell the story of a disease that shaped the modern world. By pinpointing the origin in Central Asia, scientists have not only solved a historical puzzle, but also created a foundation for future research into ancient pandemics and emerging diseases. What made the Central Asian steppe the perfect birthplace for the Black Death bacterium? How did the local environment and animal hosts contribute to the plague's deadly potential? The real geographic origin. The Central Asian steppe stretches across a vast, windswept region of grasslands, deserts, and mountain, covering parts of modern-day Kazakhstan, Kyrgyzstan, Mongolia, and western China. This expansive environment may seem harsh and empty, but it was the perfect cradle for one of history's deadliest pathogens, the bacterium Yersinia pestis, the cause of the Black Death. Why did this remote region become the birthplace of a plague that would devastate the world? The answer lies in the unique ecology of the steppe and the animals that live there. The area is home to a diverse range of wild rodents, especially species like marmots, gerbils, and ground squirrels that serve as natural reservoirs for Yersinia pestis. These animals can carry the bacterium without dying quickly, allowing it to persist and evolve within their populations. Fleas act as the disease's vector, biting infected rodents and then transmitting the bacteria to new hosts. The complex relationship between rodents, fleas, and Yersinia pestis created a stable plague ecosystem in the Central Asian steppe. This ecological balance allowed the bacterium to survive for centuries, occasionally spilling over into humans who lived nearby or passed through the region. Historical records show that plague outbreaks occurred in Central Asia long before the 14th century. Travelers and traders documented mysterious illnesses resembling plague along the Silk Road. This ancient trade route connected East Asia with Europe and the Middle East, creating the ideal pathway for the disease to spread. Caravans, horses, and merchants moving across the steppe inadvertently carried infected fleas and rodents, sparking outbreaks far from the plague's origin. The nomadic peoples of the steppe, such as the Mongols, also played a role. Their expansive empires and frequent military campaigns increased human movement over vast distances, facilitating the transmission of Yersinia pestis. It is even believed that the Mongol conquests may have accelerated the spread of the plague from Central Asia westward into Europe and the Middle East. Climate may have been another important factor. Fluctuations in temperature and rainfall affect rodent populations and flea activity, influencing how often the plague bursts into outbreaks. Warmer, wetter years could boost rodent numbers, increasing the chances of transmission to humans. These environmental dynamics created cycles of plague activity, with periods of dormancy interrupted by sudden epidemics. Recent genetic studies have traced the specific lineage of the Black Death bacterium to this Central Asian reservoir. The strain identified in ancient human remains matches the strains found in local rodent populations even today, proving a continuous presence of the pathogen in this region for over 700 years.
Now, researchers believe they've discovered the origins of the Black Death more than 600 years.
Understanding this natural plague reservoir also helps explain why the Black Death appeared so suddenly and spread so explosively. The steppe acted as a long-term incubator, allowing Yersinia pestis to gain genetic mutations that increased its ability to infect humans and spread efficiently via fleas. Once it entered human populations along trade routes, there was little to stop the rapid transmission across continents. This discovery shifts the narrative from the idea of the plague coming out of nowhere to a story of an ancient, evolving bacterium nurtured in a unique ecosystem. It highlights the interconnectedness of human history, ecology, and disease, a complex web where environmental factors, animal hosts, and human behavior combine to create a pandemic unlike any before. Implications of the new findings. The revelation that the Black Death originated in the Central Asian steppe, confirmed by ancient DNA evidence, reshapes not only our understanding of history, but also how we think about pandemics, disease evolution, and humanity's ongoing battle with infectious diseases. This breakthrough holds profound implications across multiple fields, from history and epidemiology to modern public health. First and foremost, pinpointing the real origin settles centuries of debate. For historians, this is a landmark moment. Instead of relying solely on fragmented historical records and indirect evidence, researchers now have solid genetic proof tracing the Black Death's roots. This clarity helps paint a more accurate picture of how the plague moved through ancient trade networks, linking Asia, the Middle East, and Europe. It underscores the critical role of human movement and commerce in spreading infectious diseases, a lesson just as relevant today as it was 700 years ago. For epidemiologists, these findings deepen understanding of how pathogens evolve and adapt over time. Yersinia pestis did not appear suddenly in Europe. It developed over centuries within animal reservoirs in Central Asia. Studying the genetic changes in ancient and modern strains reveals how mutations can increase a bacterium's virulence, transmission efficiency, or ability to jump between species. This knowledge is crucial for predicting and preventing future outbreaks of plague or similar zoonotic diseases, those that originate in animals before spilling over to humans. Moreover, the research highlights the importance of environmental and ecological factors in disease emergence. The steppe's unique ecosystem provided a natural reservoir for Yersinia pestis, illustrating how certain landscapes and wildlife populations can incubate dangerous pathogens. As climate change and human expansion continue to alter environments worldwide, understanding these natural reservoirs becomes vital for monitoring and controlling potential pandemics. This new understanding also influences how we approach historical pandemics beyond the Black Death. The success of ancient DNA analysis opens doors for studying other diseases like smallpox, tuberculosis, and even the 1918 influenza pandemic. By applying similar genetic detective work, scientists can trace origins, evolution, and spread patterns of many past outbreaks, offering insights that textbooks could never provide. The findings serve as a reminder of the interconnectedness of global health, Just as medieval trade routes allowed Yersinia pestis to travel vast distances, today's globalized world enables rapid spread of infectious diseases across continents. Lessons from the Black Death's origin emphasize the need for vigilant surveillance, international cooperation, and investment in disease research to prevent future catastrophes. Finally, this research has cultural and educational impacts. It corrects misconceptions about the Black Death's origins and dispels myths rooted in fear and prejudice. Understanding the scientific basis of pandemics fosters informed discussion and reduces stigma. It also honors the victims by uncovering their story with respect and rigor, connecting us more deeply to our shared human history. What challenges do scientists face when studying ancient DNA? And what mysteries still remain about the Black Death and other pandemics? Challenges and limitations of ancient DNA research. While ancient DNA analysis has revolutionized our understanding of diseases like the Black Death, the journey to these discoveries has not been without obstacles. Extracting and interpreting genetic material from remains buried for centuries presents unique challenges that scientists must carefully navigate. First, the condition of ancient DNA itself is fragile. Over time, DNA molecules break down into tiny fragments due to natural processes like heat, moisture, and microbial activity. This degradation means researchers often work with highly fragmented and damaged genetic material, which can be difficult to sequence accurately. Finding enough usable DNA in old bones or teeth requires meticulous sampling and sensitive technology. Contamination is another major concern. Modern DNA from humans handling the samples, or even from microbes in the environment, can easily mix with the ancient genetic material, leading to misleading results. To prevent this, labs dedicated to aDNA research maintain ultra-clean conditions, use protective gear, and apply strict protocols to separate genuine ancient sequences from contaminants. Even with these precautions, interpreting the data isn't always straightforward. Ancient DNA tells us a lot, but there are limits. For example, while researchers can identify the presence of Yersinia pestis and trace its genetic lineage, they can't always determine the exact way the disease spread between people or why some outbreaks were deadlier than others. Historical context and archaeological evidence must be combined with genetic data to build a fuller picture. Another challenge is the availability of well-preserved samples. Not all burial sites contain remains suitable for DNA extraction, especially in climates where preservation is poor. This limits the geographic and temporal scope of studies, sometimes leaving gaps in our understanding of how diseases evolved and spread. Despite these hurdles, advances in sequencing technology and analytical methods continue to improve the quality and quantity of ancient DNA data. Future research will likely uncover even more details about the Black Death and other pandemics, refining our understanding further. Archaeological discoveries and burial practices. Beneath fields, villages, and city streets across Europe and Asia lie silent witnesses to the Black Death's terrifying sweep. Mass graves filled with the remains of thousands of plague victims. Archaeological discoveries of these burial sites have not only illuminated how societies coped with overwhelming death, but have also become critical sources of scientific insight into the plague's true nature and origin. During the height of the Black Death in the mid-14th century, the sheer scale of mortality overwhelmed traditional burial customs. Normal graves became impossible to dig quickly enough. To contain the spread of disease and deal with the enormous death toll, communities resorted to mass burials, large pits where multiple bodies were interred simultaneously, often hastily and without individual identification. These plague pits, scattered from London to Milan and beyond, tell a chilling story. Excavations have revealed bodies buried in layers, sometimes stacked or dumped, reflecting the desperation and urgency of those times. Archaeologists have found that in many cases the dead were buried fully clothed and with no accompanying grave goods, indicating a break from typical ritualistic burials. What makes these sites invaluable to modern science is their role as time capsules preserving biological material. Teeth and bones recovered from plague pits have allowed researchers to extract ancient DNA, leading to the groundbreaking identification of Yersinia pestis as the plague's cause. Without these burial sites, uncovering the real origin of the Black Death through genetic analysis would have been impossible. Furthermore, the location and condition of these graves provide clues about the social response to the pandemic. Many mass graves are found near city walls or just outside town boundaries, illustrating efforts to quarantine the infected or keep death at bay. The placement also reflects contemporary beliefs about contamination and contagion, and how fear influenced urban planning. Archaeologists have also examined burial patterns to understand how quickly the plague moved through populations. The rapidity of mass burials indicates the swift and devastating mortality rates. In some cases, burial pits date to specific outbreak waves, helping historians map the timeline and spread of the disease. Interestingly, variations in burial practices have been observed between regions and social classes. While many common victims were consigned to mass graves, some wealthy individuals received more traditional burials, suggesting that social hierarchy persisted even in death during the plague. Beyond providing biological samples, these archaeological findings deepen our understanding of medieval society's confrontation with disaster. They highlight human resilience, adaptation, and the intersection of cultural beliefs with practical responses in a crisis. What can the ancient story of the Black Death teach us about the pandemics we face today? How do the challenges and responses from centuries ago echo in our modern world? Black Death and modern pandemics. The Black Death stands as one of the deadliest pandemics in human history, but its story doesn't exist in isolation. When we look at modern outbreaks, like the COVID-19 pandemic or Ebola, we see striking parallels and lessons that connect centuries of human experience with infectious diseases. Comparing these pandemics helps us understand how science, society, and public health responses have evolved, and why uncovering the origins of diseases remains crucial today. One of the clearest similarities is how human movement and connectivity accelerate the spread of disease. Just as the Black Death traveled along the Silk Road and maritime trade routes in the 14th century, modern diseases exploit global travel networks. Today, airplanes can carry viruses around the world in a matter of hours, much like merchant caravans once spread plague-infected fleas across continents. This rapid mobility means that understanding a disease's origin and transmission pathways is vital to controlling outbreaks before they become pandemics. Another parallel is the role of zoonotic transmission, diseases jumping from animals to humans. The Black Death's bacterium, Yersinia pestis, originated in rodent populations before infecting humans. Similarly, COVID-19 is believed to have originated from bats or other wild animals, and Ebola is known to spread from fruit bats to humans. These zoonoses underscore the importance of monitoring animal reservoirs and human-wildlife interactions to predict and prevent new diseases. Yet, the tools available to combat pandemics have transformed dramatically. In the 14th century, no one understood the causes of the Black Death or how it spread. Fear, superstition, and misinformation were rampant. Today, modern genetics and epidemiology allow scientists to rapidly identify pathogens, trace their origins, and develop vaccines and treatments. The discovery of the Black Death's origin through ancient DNA showcases how far science has come and how it continues to inform public health. The social impacts also show some echoes. Both historic and modern pandemics expose societal vulnerabilities and inequalities. The Black Death led to labor shortages that empowered peasants, but also triggered scapegoating of minorities. COVID-19 highlighted disparities in healthcare access and economic security worldwide. Understanding these patterns emphasizes the need for equitable responses to health crises. Finally, the Black Death story teaches us the importance of historical perspective. By learning how past pandemics emerged and spread, we can better prepare for the future. The insights gained from studying ancient DNA not only solve old mysteries, but also equip us with knowledge to confront new threats in an interconnected world. Ethical considerations. The groundbreaking discoveries about the Black Death's origin owe much to ancient DNA, ADNA research, where scientists extract genetic material from centuries-old human remains. However, this powerful science also raises important ethical questions. How do researchers balance the pursuit of knowledge with respect for the dead and their living descendants? What responsibilities do scientists have when handling human remains, and how do they navigate cultural sensitivities surrounding burial sites? Exploring these issues is crucial to conducting ancient DNA research responsibly and with integrity. One of the foremost ethical concerns centers on the respect owed to the individuals whose remains are studied. These are not mere samples, but once living people, often part of communities with rich cultural histories. Disturbing graves for scientific purposes can conflict with beliefs about the sanctity of the dead and the afterlife. For many descendant communities, especially indigenous groups or those with strong ancestral ties to a region, excavation and DNA analysis may feel intrusive or even sacrilegious. Therefore, obtaining permission and involving local communities in decision-making is a key ethical practice. Collaboration ensures that research honors cultural values and incorporates perspectives that scientists may not fully understand. In some cases, communities may request reburial after sampling or impose restrictions on the kinds of analyses performed. Recognizing these wishes helps build trust and demonstrates respect beyond the purely scientific realm. Another challenge is navigating the ownership and control of genetic data derived from human remains. Who owns ancient DNA? Should descendant communities have a say in how the data are used, shared, or published? These questions are increasingly relevant as genetic information can reveal sensitive details about ancestry, migration, and identity. Transparency about research goals and data handling, as well as sharing benefits with affected communities, promotes ethical stewardship. Balancing scientific inquiry with cultural sensitivity also means confronting difficult choices. The pursuit of knowledge about historic pandemics or human evolution can conflict with the need to preserve burial sites intact. Researchers must weigh the potential benefits of DNA analysis, such as understanding disease origins or helping modern medicine, against the possible disruption to sacred sites. In many cases, non-invasive or minimally destructive methods are preferred, and when excavation is necessary, it is done with utmost care. Legal frameworks around ancient DNA research vary by country and often reflect local customs and histories. Regulations may dictate how remains can be handled, where they can be studied, and how findings should be reported. Adhering to these laws is fundamental, but ethical responsibility extends beyond legal compliance to fostering respectful, meaningful dialogue with stakeholders. Moreover, ethical considerations include the communication of research findings. Scientists must avoid sensationalism or misrepresentation, especially when dealing with topics as sensitive as pandemics and human suffering. Presenting results in a way that honors the dignity of past populations and avoids stigmatizing any group is essential. The case of the Black Death's genetic research exemplifies these challenges and the progress made. Many studies involved international teams working closely with local archaeologists and governments. This collaboration ensured that sampling was conducted ethically, with appropriate permissions and cultural awareness. The outcome has been not only groundbreaking science, but also a model for respectful interdisciplinary work. As ancient DNA research expands, it faces evolving ethical questions. Advances in technology enable more detailed analyses, sometimes revealing unexpected or controversial information about populations and their histories. Ongoing dialogue between scientists, ethicists, and communities is necessary to navigate these complexities. Ancient DNA research sits at the crossroads of science, history, and culture. Its ability to unlock secrets of the past must be matched by a commitment to respect and responsibility. Ethical considerations are not obstacles, but guiding principles that enrich the research process, ensuring that discoveries honor both the dead and the living. Have you ever heard about Black Death from your grandparents? Write your stories below. Subscribe for more uploads in this channel.